new courses, and there's some little tweaks here. So just a few comments. We're redoing some of the prerequisites for these courses. For instance, a lot of students don't take biochemistry because it's really an elective and such. Since this is a university, you would think I could be able to just have the prerequisite deleted, but since it's a university, it's going to take about a year to go through the paperwork and all the numerous committees and such. So we have to just do overrides on these things. So Dr. Gia has said that if you're interested in his molecular genetics of aging course, but you haven't taken the molecular and cell biology, just send him an email and he'll give you an override, and so you can get into that course. For the biotech lab, formally, it's still in the catalog. We're changing things around a little bit. It's got a requirement for general micro and biochemistry. Really, the main course you must have to be able to make any sense out of the course at all is the one you're doing right now. So if you like genetics, if you think that you'd like to, let's say, work for Scripps or Max Planck as part of a research group, these courses are really intended to be very hands-on, give you the real skills that are actually used in the laboratory. So we do basic manipulations with DNA, protein, and RNA. How many of you have taken the organic lab? All right. So you still write those long, horrendous 20-page lab reports? Is that right? Okay. In biotech, we don't do that because I'm not going to read that many pages. Okay. The lab reports are more bioinformatics assignments, which are going to be similar to some of the topics we'll get into, teaching you how to get into the databases and such. The other complication to it is Biotech 2 requires Biotech 1 as a prerequisite. It's not always clear in the limitations of the Banner system. These are so-called fast-track courses. So you'll sign up for both courses in the same semester. So it's actually 4 credits worth of lab courses. We do all of Biotech 1 in the first half of the semester. We go to spring break, we come back, and we do all of Biotech 2. So if you can get signed into Biotech 1 and don't need an override, then just send me an email that says I signed up for Biotech 1, but I can't get into Biotech 2, and I can give you the override that way. So we'll get all of that fixed up. Genes in Development has turned into a very popular course over the couple of years that it's been offered. Right now, I just talked to Dr. Trevigno. She's fine with raising the cap on it, another 15 students. I'll be 15? No, maybe another 4. I think it's at 100 now. We're going to try to raise it up to 140. Evolution. This one is a very popular course, and for some administrative reasons, we had to keep the size of the current section being offered by Dr. Frazier at 125. We're working with Dr. Hughes right now. We're going to try and add another section that'll meet on a Wednesday, Friday. Yes? In Dr. Frazier's section? I'm not sure. The other is when I get the second section up, some other students may transfer out of hers because it fits their schedule better. So, I mean, we're still almost 2 months away from the beginning of class. So we'll just have to see how it plays out. Practical cell neuroscience. This is if you're interested in neurobiology, and there's kind of a comment running around through the scientific community that within the next 5 years, every other scientist will be a neurobiologist. Or if you go to the national meetings now, I think they're up to like 15,000 people attend these national meetings. This practical cell neuroscience with Dr. Dawson Scully, it's a computer simulation, so it's all done in a computer lab. It's a lecture lab, but it gives you the basics understanding so that you can do the next lab, which is really the wet lab with all the electrophysiology equipment and such. The Boca sections are filled, but it's being offered in both Boca and Jupiter. So my comment here is the lecture is on Monday morning, but there's a lab on Monday and a lab on Tuesday. If it works out with your schedule, if you're going to Jupiter for another class or whatever, you can sign up for the Jupiter section because there's seats that are open. Let's say, but you're going to be in Boca on Monday, you can come to the lecture in Boca on Monday, but take the lab in Jupiter on Tuesday. So if that's a little confusing or doesn't make sense, after class, come on down and I can give you all the details because I've thought about this and dealt with it way more than I want to admit to. And a new course, Molecular Genetics, is actually an old course that I put in the catalog way back in the dark ages when I first arrived at FAU. Hasn't been used for many years, and now it's being brought back in a new form. But it still has these old general microbiology and biochemistry prerequisites. So if you're interested in the molecular genetics, the molecular side of what we're doing, I got an email exchange with Dr. Thiessen this morning. If you're at least on track to get a C in genetics, he'll waive the other requirements. Now, be aware, don't lie to him because he will email me and want to know if you really are getting it. If you're not getting at least a C in here, don't sign up for it. You're not going to be happy. Yes? The main ones here, yeah, the general biochemistry is helpful, but really it's the genetics. So I think if you have an interest in it, you like the genetics, then I'm fine with overriding these two prerequisites. Yes? I would just keep looking at it because I never even know about it until it just pops up on Banner. I submit the request to add it, and then it goes off into the ether somewhere, and they don't even tell me. So just keep, yeah, in terms of any of these, just keep an eye on the Banner system. Once scheduling actually puts it into Banner, it goes up in real time. But I'm hoping to see if I can get all that squared away this week. Yes? The course number? Not exactly, but it's a BSc, so it's up near the top. It's like, it should be, I think, yeah, it's a BSc 4000 and something. Okay, so if you have any other questions about the details of the schedule, I can probably answer them or what's coming. It's kind of like insider trading. I know kind of what's going on. And so after class, we'll have time. If you want to come down, you can just ask me questions. Okay. What's probably going to come as a big disappointment to many of you, but after today, we only have 2 lectures left. See, I knew a lot of groans would go out, but that's okay. You know, all good things have to come to an end at some point. I'm hearing some of that. I have selective hearing loss. Okay. So let me make a couple of things clear here. I sent out the email that said that I was going to omit Chapter 19. Basically, I had to give up something because we have one less lecture period than I had last fall because both Veterans and Labor Day fell on a lecture class. So I've chosen to give up Chapter 19, which is on the technology, which is the really cool stuff, which is like, you know, the stuff you get to play with in the lab and what's fun to do. But a lot of that, it's hard to make sense out of it unless you actually are in the lab doing it. And the other is the material that I'm going to cover in the last 3 lectures, including today, after we get out of the DNA repair chapter, is really what I would lump under modern genetics. This is really where we are going today. So we've gone the whole route from Mendel all the way up to present day genetics, and we'll talk a little bit about personalized medicine and bioinformatics and these kind of areas. These are complicated areas. They are the kind of topics that entire courses can be built around. So it's going to have to be a very kind of broad brush overview to things. We're not going to be able to get into the details of it because there's just not enough time. The idea is for you to see whether you really have if there's a basal interest there that wants you to, so that you want to know more about this. You like the developmental genetics when we get to it, then take genes and development, these sorts of things. I was surprised at how many emails I've gotten with students wanting to know whether I was just dropping Chapter 19 from the lecture, but they would still have to know it for the exam. Have I asked you to learn anything on your own? No. Why would I start now? Okay. So it will only, if we didn't cover it in class, it's not on the exam. The only caveat to this is maybe at the end in that last lecture, if there's a little bit of time, I might covered just a couple of the really sort of cutting edge techniques out of that chapter 19. I'll let you know about this. It'll be very limited. We may not even have time to do it. But we're going to, for instance, there'll be a little bit of it splashed in through some of the lectures that we are going to cover over these last three lecture periods. All right. So with that introduction, let's do our two questions. So this one should be from the last lecture, which I know most of you are reading about over the weekend, so it should be pretty easy. Whoops, come on. There we go. This is going pretty fast, so I think 1 minute should be fine. All right, let's see. Okay. And the answer is what? Uracil, sure. Oops. Okay, and in a few minutes we'll talk about how cells recognize that uracil is present in the DNA. It's highly mutagenic, and how they repair it. And then for where we're going today, or something that I've mentioned numerous times throughout the course. Can you tell me how many genes in the human genome, at least to the closest order of magnitude? Oh. All right, we'll go to one. Most of you are getting this one too, so we'll go to 1 minute on this. Two easy ones here to get the day started. All right. And what's the answer? Okay. So, getting to the actual material we want to cover. So we've been in this long chapter on DNA mutations and repair. The first part of it talked about the different types of mutations, some of the classic kind of mutations that you know about in terms of single base mutations, point mutations, nonsense mutations, and such. The second part was how those mutations can happen, whether through chemical reactions such as the deamination reaction we saw before, or cosmic radiation, all these different types of areas. And so the last part is really a little introduction into how cells recognize and repair that damage. But before we finish off the chapter with that, where we stopped last time was a discussion of transposable elements, which are themselves they are mutagenic. So these, we went through a few of these slides. We'll just kind of start here to get back. So most of the time, people call these transposons. That's probably the most common name now. They've been called mobile elements, jumping genes. What else here? Controlling elements. That's probably from McClintock's work. They all mean the same thing. But as it says here, probably transposons and transposable elements are the most common terms. So it turns out they're far more ubiquitous and abundant than people ever imagined. So we talked about some of those landmark experiments right in the late 1940s, early 1950s that talked about that provided conclusive evidence that genetic material was encoded in DNA, not protein. So, okay, the scientific community is okay with that, but they kind of viewed DNA as this monolithic structure that occasionally had a little mutation here or there. And then this crazy lady comes along, her name is Barbara McClintock, and who was arguably the best maize geneticist in the world at the time, and was working on what's maize is corn. So she was both a cytologist, which meant she was an expert at examining the chromosomes of corn under the microscope, and a corn geneticist. And she said, well, you know, based on my experiments, there's parts of this DNA, these genes, they're moving around. And no one really understood what she was talking about at the time. They didn't have a good context. They were just beginning to accept that DNA was actually an interesting molecule. So in a lot of ways, like Mendel's work, her work was pretty much ignored for probably all the way into maybe the 1970s or so, when these jumping genes, these transposable elements, were found in E. coli. And then people started realizing maybe McClintock was onto something. And now, to my knowledge, every organism that's been examined, including humans, there are transposons. They are ubiquitous. McClintock did win the Nobel Prize for her work, and she was working at Cold Spring Harbor until shortly before she passed away. So she was working well into her 80s, probably close to 90 years old. We talked about this junk DNA, which is now sort of thought about in a completely different context. But when you look at the structure of it, and we talked about how there's all these repeated DNA sequences, about half of that repetitive DNA looks like it's leftover pieces of transposons. So they're scattered all through the genome. There's many, many copies of them. Most of them have now lost their ability to transpose, to move from one place to another. So they just kind of like, you know, that guy who's collecting all those Chevys has them up on blocks in his front yard. None of them run, but you can tell what they used to be, kind of thing. They are mutagenic. We talked about this. I kind of gave you the analogy with the movie with the Terminator and Snow White and such. So they are segments of DNA. For instance, I'll tell you about one in detail, the P element from Drosophila. It's about 3,000 base pairs of DNA. And as far as we can tell, they can pretty much insert themselves into the chromosome wherever they want. So if they insert in the middle of the gene or in the promoter and such, they clearly can disrupt the entire function of the gene. Also, many of these transposable elements are present in multiple copies within the cell, and since they're homologous, they promote genetic recombination, so they can lead to insertions, deletions, all sorts of chromosome rearrangements. So they should be very, very mutagenic in genomes. There's an incredible range of these, diversity of these transposable elements. In Drosophila alone, well over 30 different classes of transposons have been found. I think people just gave up trying to categorize them at that point. There were just so many of them. But they do share a couple of common features. And here's what we were talking about as we kind of came to the end of the lecture last time. At the ends of the transposons are an inverted repeat. So they're part of the transposon. That inverted repeat is necessary for the mechanism for inserting into the DNA. And I'll show you how that happens in a minute. But then what you find flanking, if you're sequencing the DNA and you see these inverted repeats, directly, or let's say flanking them right next to those inverted repeats are direct repeat. And that direct repeat is actually the result of, those direct repeats are not part of the transposon. They are from the genome, and they are a result of this insertion mechanism. So you'll see this kind of inverted repeat, direct repeat. And so if I show you in a moment how this mechanism works, you'll see how they arise. They also need an enzyme called a transposase. So this transposase works with these inverted repeats. So in order for for most of these transposons to jump around from chromosome to chromosome or wherever they're going to move throughout the cell, they need both the transposase and the inverted repeat. So this P element I'm going to tell you about in some detail. One modification of it has been called wings clipped. they removed the inverted repeats on the end. The transposon can insert itself, but it can't come out again. So but that gets into the manipulation of how it was used for experimental purposes. So here's sort of what you see when you have a transposable element inserted into the genomic DNA. We have this direct repeat, which is the sequence reads the same on the same strand in the same polarity. All right, so that one's pretty easy to recognize. And then our old friend, the inverted repeat, is next to it. Everybody see how it is an inverted repeat? You know, so this is part of the transposon, or the transposable element. This part comes from the chromosome. So that's the result of where it inserts. The reason it works that way is this enzyme, this transposase that I told you about, makes so-called staggered cuts. What that means in the jargon is where these arrows are pointing, the enzyme cleaves the phosphodiester bond between those two bases. So the only thing that holds it together is the hydrogen bonding between, what do we got? Looks like, well, we got 5 bases there. 2, 3, 4, 5. All right, look, yeah, or 6, no 5. So it's cutting here and up here, and the only thing holding the two strands together now is the hydrogen bonds between these 5 bases. And physiological conditions are going to break, come apart, and you create these so-called sticky ends. So if you know a little bit about restriction endonucleases, this is very analogous to that. Then the transposable element inserts itself, but now we have these gaps in the DNA which have to be repaired, and so the DNA polymerase comes in, fills in those sequences, and that's how those direct repeats are formed. They're a direct consequence of the mechanism by which the transposase inserts. If the transposase didn't make stagger cuts, in fact, if it cut a DNA sequence at the same site on both strands, would you have direct repeats? No. So, and the direct repeat will be the length of basically where, well, I won't say, it just makes it confusing. Okay. So it's because of the staggered cuts and the way the transposon inserts and then the repair of these gaps that the DNA actually gained 5 nucleotides in sequence, and it gives you these direct repeats. Okay. Any questions about how that works? The other thing is the copy number. Some of these transposons replicate by a mechanism called replicative transposition, which means simply a new copy of the transposon is made. It goes and inserts somewhere else, but the original stays where it is. So now you can have numerous copies of the transposon within the same cell. Whereas a non-replicative transposon comes out from one site and goes to another one. But then most likely you're only going to find one copy of the transposon. So these are just 2 general mechanisms by which they replicate. We talked a little earlier about this mutagenic effect, and there's a lot of cases where we know that, in fact, the mutation we're looking at is the result of a transposon. If you remember, we talked about the bar mutation. So this was, we talked about gene duplication, and this was that shape of the eye in Drosophila, and the more copies you had, the more extreme the phenotype. Well, in fact, that mutation is the result of recombination between 2 transposable elements. So it is a transposable element-driven mutation that occurred. Okay. So let's try this question. I added this just before class, so hopefully it makes sense. You get to edit it. We'll go to 1 minute and 15 seconds on this one. Let's see. Okay, well, we'll go to 1:30 since I had my back turned. All right. So what's the answer? It needs a little bit of editing, but okay. So the whole purpose was you have multiple the idea of the question was you have 10 copies of the transposon. The inverted repeat is part of the transposon, so it will be the same on all 10 copies. But the direct repeat is the result of where it integrates into the chromosome. So the direct repeats would be different. As far as we can tell with most transposable elements, they don't have a specific DNA sequence that they integrate at. It just appears to be very much like the F factor integrating into the chromosome. Oh, there we go with that question again. It appears to, as far as we can tell, it appears to be random. Okay. So as I said, there's been more than 30 different classes of transposable elements that have been found in Drosophila. Let's see. Some of them have some interesting. The first one found was called copia because it was found because an mRNA transcription from one of the transposon genes was present in such high concentrations in the cell. Others have been called, I think there's one called Jiminy Cricket, the HMS Beagle, Gypsy. It's one way I like Drosophila genetics. There's a lot of times there's a backstory as to why the genes were named. You know, the yeast geneticists are more like some kind of an English banker. You know, very like the first transposable element was called TY1 for transposon in yeast number 1. Not very creative in terms of the naming. Okay. Well, one particular P element that's been used extensively, and it's really been developed into a very powerful tool for molecular geneticists working with Drosophila, is a transposon called the P element. And most of the wild-type P elements are almost 3,000 base pairs. They're about 2,900 base pairs in length. Now, as I said, this has been used extensively as tools now, so this P element has been heavily modified for various applications that are used in the genetics, but it's still, just like we had before, it has a 31 base pair inverted repeat. As I said before, if you remove these repeats to get what was called wings clipped, the P element can integrate into the chromosome, but then it becomes stabilized and it can't excise back out of the chromosome. And then it has an 8 base pair direct repeat that's based on its insertion site. It's got kind of an interesting molecular biology behind it. So, based on its natural biology of what's going on here, it was the discovery of something called hybrid dysgenesis. So this was in Margaret Kidwell's lab at Brown University, and they were going out and finding flies out in the wild and bringing them back, and they were probably doing some population genetics and mating them with lab strains. And they found a really interesting phenomenon. If they took female flies from the wild, they went down to the local fruit stand and started picking them off of the bananas. I'll think of something in a minute. What is it? Time flies like an arrow, but flies like a banana, something like that. Anyway, I don't know. I think I read that on a bathroom wall in the genetics lab once. So you get females from the wild, you bring them in, you start mating them with the males that from lab strains that have been carried for years in the laboratory, and the F1 progeny are fine, and the F2 generation looks fine, and there doesn't seem to be anything going on. However, if you take males that you caught from the wild and you do the reverse, you bring these wild males into the lab, mate them with the female strains that have been carried in the lab for a long time, you find that either your F1 progeny are sterile, or if they're not sterile, the F2s have a very high level of mutation, and all sorts of chromosome aberrations and such. And they didn't really know what was going on here. So it really depended on which way they did the cross. Well, it turns out what happens is, let's see if this is the one I'm looking for. All right. So when you bring in females, what they're bringing is bringing in females from the wild that had this P element transposon, but they also made a repressor so that in the eggs, while the P element was there was a repressor protein that prevented the transposon from moving. So it wouldn't be mutagenic. It really was going to cause mutations once it started moving around. And so they crossed that with these males, and not a problem. What happened was, what was causing the dysgenesis is the males also had the P element. But what they found was the lab strains, after these females have been carried generation after generation for many years in the lab, they lost the P element and the repressor. So as soon as they crossed them to these males that had the P element, then they started without the repressor, and because it was the P element, you started to get transposition, and the P element starts jumping around, causes mutations in the chromosomes. But the interesting thing was that the F1 progeny looked fine, but they were often sterile. Or if they weren't sterile, then the F2 had all sorts of phenotypic aberrations and such. The reason was the transposon was only jumping around in the germline cells, not in the somatic cells. The reason for that was the result of some differential splicing. Okay. So the reason they saw this dysgenesis, that in other words it depended on which way they did the mating, was because transposition only occurred in the germline cells. Okay. Makes sense? All right. So, and it was again a result of, it wasn't the transposase was only made in the germline cells. The gene was expressed, but just like we've talked about before with the sex lethal, it was a result of differential splicing, so that only a functional transposase was made in the germline cells. Okay, so that's what all of that says. That's what all of that says. Okay. So we look a little bit at the human transposons, and we're not going to go into it because we've talked about this before. They are the source of a lot of this repetitive DNA sequence that we carry in our genomes. As I said, it looks like they're sort of derelict transposons that have lost the transposase, or maybe they've lost the inverted repeat, so that they no longer can move around, and they're just pieces of DNA sitting there, which for many, many years was thought to be junk DNA. And as I've said numerous times, we're changing our ideas on that now. Okay. And so this kind of gets into the biological question is, why are there transposons? In other words, some people think they really have an evolutionary role. They provide, as a source of mutation, they provide new alleles. You can test out new alleles in new environments, see if there's some sort of selective advantage. Other people have called them selfish DNA because they just seem to care about themselves and nobody else. And right now, there's just no clear answer. So either one of those answers would be correct at the present time. So we got something we know a lot about. We know it's ubiquitous, but we really don't have a clear idea of what the real function is, if there is any at all. Okay. All right. So let's try this question about the transposons. Details. It's always the details. Let's try. There we go. She switches out the battery. 1 minute should be good on this one. She can get her batteries changed in less than a minute. This is resourcefulness. You guys doing swapping out batteries?
I usually carry a spare.
All right. There you go. For those who are paying attention, I hit the wrong button. All right. So I didn't hear the usual uproar of giggles as half the class missed it. There's 3 types of people: people who make things happen, people who watch things happen, and the rest who go, "What happened?" Okay. So the last part of this is the repair mechanisms. I'm just going to give you, I think it's just 3 examples of DNA repair systems from E. coli, show you how they work. This is just the tip of the iceberg. There's many, many more DNA repair systems. But except for the one example I'm going to give you, they all share in common. The idea is identify the damaged DNA, and then you remove the damaged DNA and a little bit extra, and then replace it with new DNA. So I've used the analogy, and I've mentioned it before, that you got a wooden porch on the back of your house, and the one board's a little rotted. So the first thing is, how do you identify it? Well, when your foot goes through it, you've identified where the damage is. Then you take the old rotten board out, you put in a new board, and you're good to go. So the key here is with the DNA repair systems is how do they find the lesion? And then, except for the one example I gave you, then it's remove the old DNA along with some new DNA, just replace it with new DNA, and the damage is fixed. So the key to this is really what type of lesions can each system recognize and repair. One of the first ones that's been worked out in a lot of detail and very, very well known is the mismatch repair system. So way back when we were talking about DNA replication, we talked about how DNA replication occurs at such a high level of fidelity that in many systems, no more than 1 in a billion nucleotides ends up being incorrectly inserted during DNA synthesis, but that there are no enzyme systems that have that level of fidelity. And it really comes from proofreading repetitively. So we talked about the proofreading activity associated with the exonuclease of the polymerase, but then the question is, that system's not perfect. So what happens to mismatched base pairs that escape that checking by the polymerase and are actually present after the DNA is synthesized? So this is a post-synthetic repair system. The DNA is already double-stranded, and now there's still a mismatch, and so this mismatch repair system goes in and corrects that problem. Okay. So here's the key problem. The DNA is double-stranded at this point. How does the repair system know which one is the correct base and which one is the incorrect base? Or does it just take a guess, repair one at random, and half the time gets it right and half the time gets it wrong? So how can this repair system recognize which of the 2 DNA strands has the wrong base? Well, this is our old friend methylation. So there's an enzyme called the DAM methylase for deoxyadenosine methylase. And in bacteria, so we're looking at an E. coli system here, methylation nearly always occurs on A sequences. So what this DAM methylase does is on when there's a GATC sequence, it methylates the A. Now, this is why this repair system can work. This methylation occurs after DNA synthesis. So there's a window of time before the methylase methylates the new DNA where the DNA is only methylated on one strand. So we call that hemimethylated. It's just methylated on one strand. And so if the repair system can get to the DNA mismatch before the methylase acts on the new DNA, it can tell which one's going to have the correct base? The methylated or unmethylated? The unmethylated will have the incorrect base because the methylation is from the old DNA that was used as the template strand. So all it has to do is find the methylated DNA and remove the base on the opposite strand. So the system is called MUT-SHL. So these were 3 genes, MUT-S, MUT-H, and MUT-L. They got their names because mutations in any one of these 3 genes resulted in higher levels of mutagenesis. So that's how it got the name MUT, for increased mutations. The 3 proteins work together as a complex. And what they're really going to be doing is they're going to find the mismatch, look for the closest GAT sequence, see which A residue is methylated, and then remove the mismatch base on the opposite strand. So so the lower strand is the template strand. Here's the methyl group on the A residue. Here's the mismatch. So MUT-H comes in, recognizes the methylated strand here. It's an endonuclease, which means it's going to cut internally in the DNA, and it nicks, right next to the G, it nicks the DNA on the unmethylated strand. That's the whole key right there. All right. And then MUT-S and MUT-L looks like it might not be in the picture. Well, what the key here is, here's the mismatch. Oh, this is probably MUT-L here. Maybe it's just not marked. So these 3 proteins are working together. MUT-S has identified the mismatch. MUT-H has nicked the DNA. Then exonucleases can remove that strand coming around past where the lesion was and now generating a 3' end. And then the DNA polymerase can just like it did with repairing and linking up the Okazaki fragments, the DNA polymerase comes back around, fills it all in, and the mismatch is repaired. But it has to do this in a very short window of time. Once the methylase methylates the new DNA sequence, then the repair system can't tell which is the old DNA and which is the new DNA. Okay. All right. So let's see how much of that you've retained. All right. This one's pretty straightforward. I think 1 minute ought to be plenty here. All right. So what's the answer? Okay. Yes.
So what happened to the new strand that was taken off of the genes?
The one that was degraded?
Oh, it degrades?
Yeah, so what happens is this nick occurs, and just like we talked about Okazaki fragments, basically DNA polymerase 1 can remove this, or it could even be another enzyme. Basically all this, I guess what is this, the red strand? This gets removed and cut back past, right here is where the mismatch was. So all it has to do is go past that. So now so this is the damage plus some good DNA has been removed. And now from this 3' end, DNA polymerase 1 can come around, fills all this in. DNA ligase joins it together, and you have one solid piece of DNA again.
So is that one better than the previous?
I'm sorry, what?
Like, there's no mistakes on that one now? On the new
Right, because it's, yes, it might make a mistake again, but maybe if this is 99% correct, then this is only going to fail maybe 1% of the time, probably even less than that. It's really between these multiple levels of proofreading how you get to that point where you typically only have one mistake in a billion nucleotides. Okay. But once this new strand of DNA is methylated, then the cell really has no mechanism for finding the correct mismatch base. Okay. The second example is the one example I'll give you where the DNA, the damaged DNA is not being removed. Rather, it's being basically chemically or enzymatically, the damage is going to be reversed. So this is works most efficiently and is most often used with the formation of the thymine dimers that form from ultraviolet light. So it's this covalent bonding between two adjacent thymines as a result of absorbing ultraviolet radiation. And this is a process called photoreactivation. It got its name because it's really a two-step process. The first part of the reaction can occur in the dark, but the second part requires a photon of light. How many of you taken the micro lab? Okay, do we still do that? I think we still do the UV mutagenesis experiment. Okay, and you do it in the dark? Or don't do it in the dark anymore? Not keeping you in the dark, I'm saying the experiment's done in the dark. You don't remember? Okay. Yeah, or part of it's covered? Right. Well, that's to give you the control, but you usually do the recovery in the dark because you're trying to prevent the photoreactivation. So the whole purpose of that experiment is to show the DNA damaging capability of the ultraviolet light. You probably use increasing dosages of it, but you do it in the dark to prevent this repair by photoreactivation. So the key here is, and this is the one example I'm giving you, where this enzyme simply breaks the covalent bonds between the adjacent dimers. So the original bases are still there, it's just the damage has been repaired. In eukaryotes, it's called photolyase, but it's basically the same reaction. So it's a chemical reversal of the damage. And the last one, nope, I think I may have two left, maybe there are four. This one's, okay, stay with me. This one's going to look complicated, but it really isn't. It just involves two steps. So we talked about how you can have the deamination of cytosine, you form uracil. Uracil in DNA is highly mutagenic. It needs to be removed. The way cells do this, it's a two-step process. The first is the use of an enzyme called a DNA glycosylase. All right. All that means is it breaks the glycolytic bond in DNA. Where's the glycolytic bond? Well, the sugar phosphate, that's phosphodiester. The glycolytic bond is the bond that joins the base to the 1' carbon. All right. So it's going to cleave that glycolytic bond between the base and the sugar so that it can release. You've got a damaged base. The cytosine has been deaminated. Now it's uracil. You can just clip the glycolytic bond. The base is lost, but the sugar is still there. So the sugar phosphate backbone is still in place. And then you have to replace that missing DNA base. So I'll show you how this works in two steps. So DNA glycosylases are a family of enzymes. The one we're going to look at is called uracil glycosylase because it specifically looks for uracil in the DNA and only repairs uracil. And if it sees a uracil base, then it cleaves the glycolytic bond. When it does that, you have the sugar is still in place. You got the sugar phosphate backbone, but now there's basically a hole in the DNA. The base is gone. And so this is called an AP site for apurinic, or less commonly apyrimidinic. It just means the sugar is there, but the glycolytic bond's been broken and the base is missing. So there's really a base on one strand without any other base on the opposite strand, base paired with it. All right. Then exonucleases, polymerases, and such come in. So I think it'll be easier to show you how this works stepwise. So here's the damaged base right here. So this is, let's say, a cytosine that was deaminated. So now it's uracil. The DNA glycosylase is going to break this bond right here, the glycolytic bond that holds the base to the sugar. So that's what's coming out. And now you create this AP site. The sugar phosphate backbone is still intact, but there's a base missing. Basically, I call it a hole in the DNA. The next step is an enzyme called an AP endonuclease. All that means is it's an endonuclease that's looking for an AP site. The endonuclease will clip or make a nick in the phosphodiester bond in the sugar phosphate backbone. So it's going to cut right here, and you got to notice the orientation. I was playing around with the effects. Okay. It needs to clip it here so that there's a 3' hydroxyl. Why is that important? Because now the DNA polymerase 1 needs to remove this DNA and replace it with new DNA, and so it has to start from a 3' hydroxyl. So now, the whole setup was the two enzymes. The first, the glycosylase that clips the glycolytic bond, the base is gone. The AP endonuclease looks for these missing bases, cleaves the DNA, sets it up so that now DNA polymerase and ligase can come in, remove the damaged DNA, fill it in with new DNA, and the repair is finished. Let's see. So that's what it shows you right here. So we got DNA polymerase, fills everything in, and then just like we had to do with the Okazaki, it's very much like the Okazaki fragments. Then you need DNA ligase to form the last phosphodiester bond, and the DNA has been repaired. So that common theme of identify the damage, but the repair process is very similar for nearly all of these systems. Okay. And the last one is nucleotide excision repair. This is an important mechanism because it recognizes a wide range of lesions in DNA. So there's all sorts of different types of damage that can occur to DNA. What this system's doing is it's really just looking for anything that's out of the ordinary. It's looking at the three-dimensional topology of the DNA. If there's something irregular about that, then basically the safest thing to do is just cut it out, get rid of it, put in new DNA, and repair it that way. This one is a little different than the ones I've shown you before because it's actually going to cut the DNA twice. But I'll show you how that works in a moment. Okay. So I'll just kind of walk you through it, then I'll show you in a sort of a cartoon form. So the enzyme complex identifies the damaged DNA. First thing it does is begins to unwind the DNA. And then we have our old friends, the single-strand binding proteins. Remember we talked about those back with DNA replication. Their function is to bind to the single-stranded DNA to keep the helix from reforming. In this case, the enzyme complex cuts the DNA twice, once on each side of the damage. How far away from the damage really depends on whether it's prokaryotic or eukaryotic. So in prokaryotes, it's 3 nucleotides on the 3' side and 8 on the 5' side, or it's a little bit more in eukaryotes. But the key point is it's going to cut on both sides. Now you got a short fragment, including the damage, will dissociate because it's only being held by a limited number of hydrogen bonds. DNA polymerase and ligase come in, fill in the gap, and we're repaired. So here's the damaged DNA. Like I said, this can be a wide range of different types of lesions. The complex recognizes it, unwinds the DNA. Here's the single-strand binding proteins to keep it single-stranded. Then it gets clipped on each side, and the segment of DNA that contains the damage is removed. DNA polymerase The polymerase is going to go left to right or right to left? Who said right to left? Okay. Make sure you get that fixed before you get to the next exam. Okay. So it's going to go from the 3' end. The nucleotides always add to the 3' end. Okay. You fill it in, ligase closes it up, and it's fixed. All right. Any questions? Okay. So those are from E. coli. Similar systems work in eukaryotes, but as always, they're much more complicated. Takes many more enzymes to do the same job. Okay. So the last comments are the correlation between different genetic diseases and mutations in DNA repair systems. So there's a very clear correlation between mutations in specific DNA repair systems and increased incidences of cancer. So, for instance, one very well-studied system is a genetic condition called xeroderma pigmentosum, or it's oftentimes just called XP. These individuals are highly susceptible to skin cancers because they have a defect in the excision repair system. They can't repair the damage caused by ultraviolet light. So what you see is this very clear linkage between increased incidence of skin cancer and this inability to repair the types of lesions caused by ultraviolet light. There's lots of other ones. There's another one, ataxia telangiectasia, which is the linkage, the problem there. It's a neurological disorder. If I remember correctly, the mice that have AT, you hold them by the tail, they just start spinning around, kind of thing. No, I do not abuse small animals. I have 3 animals at home. It's the experimental model, okay? Back to the serious side. This is a defect in the ability to repair cross-linked DNA. We haven't talked about that, but there's chemicals that covalently cross-link the DNA so that it can't unwind. There's a human disease called Fanconi's anemia, another, again, defect in the ability to repair cross-linked DNA. Also, the last slide I had here, there's a hereditary form of colon cancer. This is responsible for about 15% of the colon cancer cases. I don't have time to go through it, but it's a really nice example of how studies that came from E. coli and yeast and Drosophila kind of converged together, and then along with pedigree analysis, all converged around the finding that individuals who have this colon cancer, in most cases, end up having a defect in a key gene as part of the mismatch repair system. So a lot of this came about from a large pedigree of a large family where about 50% of the individuals had the colon cancer. Every single one of them had the same mutant allele in this DNA repair system. All of the individuals who didn't have the cancer had the normal wild type allele. Okay. So the take-home message is a very clear correlation between the inability to repair DNA and increased incidences of different genetic diseases, especially cancer. Okay. So we'll use this question. All right, this one should only take a minute too. All right, and the answer is what? So So any questions on that long chapter? So, yeah, this will, so this will be this just reminded me, so I will try to remember very quickly to post up the sample questions. Probably I'll find that there's, a little bit disproportionate weighting towards this chapter because it's pretty long. But it's an important chapter, and I think it's an area of DNA metabolism that tends not to be covered in other courses. Okay. Yes?
You said that uracil is highly mutagenic.
I'm sorry, what?
That uracil is highly mutagenic.
Yes. Yeah. Yeah, the uracil, it leads to a lot of mispairing problems and replication errors. So it needs to be removed before DNA replication. Yes?
These repairs are being done by DNA polymerase 1.
In E. coli, yeah. So mostly when it says DNA polymerase, most of these, since they involve, many of these repair systems involve removing DNA and replacing it, just like you have with the Okazaki fragments. So yes, DNA polymerase 1 in these E. coli systems is usually the polymerase that's involved in this. So it's another one of these examples where a lot of these enzymes function in multiple capacities in both genetic recombination, DNA repair, and replication. Okay. So as I said, we're going to skip chapter 19 for now. We'll move into chapter 20 on genomics. This particular chapter is not going to follow the textbook. The textbook has got an incredible amount of information in it. I couldn't possibly do that chapter in one lecture. Plus, there's a level of detail in there that, let's put it this way, Dr. Narayan's got, well, it's already filled, but he's got a whole semester on bioinformatics now. But this will really try to give you an introduction to modern genetics. So, at least for me, one of the major watersheds in genetics came in the 1970s with the development of molecular cloning technology. This gave us tools that let us start to work in eukaryotic systems. Prior to that, we were pretty much limited to E. coli and viruses. And then the next was the Human Genome Project that really opened up genomics, and with advancements in the internet and computers and the speed of sequencing. This is really, so for your generation, it's really genomics is what's the driving new area of modern genetics. So I'm just going to try to give you a broad overview of some areas of how it's impacting both professionally if you work in this business and also just your everyday life as a good citizen who might, maybe you get called in for jury duty, and a lot of times, have DNA evidence now. I never got called for jury duty before I moved to Palm Beach. I think I've been called 3 or 4 times. My wife's always going, now don't get called for the jury. So but there's this thing called contempt of court, so you don't want to be too much of a smart ass. And then somebody said, no, they never take university professors because you guys ask too many questions and you're too detailed and such. But every time I've been interviewed, I've been chosen. So, like I was on one, it was kind of crazy. It was some sort of argument over the cost of some tax or the cost of some property between what the state was going to have to pay for this right of way. And the lawyer, so during that interview process, asked me if I'd ever, been involved in any real estate transactions. I said, yeah, I bought a couple of houses. I said, I find it incredibly boring. I thought that might get me off. He goes, yeah, I do too. Yeah, we'll keep him. All right. So, this Human Genome Project, which started in 1990 and was actually completed 2 years ahead of schedule. It was kind of an interesting story involving Craig Venters as far as that goes. Really initiated in this whole area of genomics. And as I mentioned a number of times, and in the first slide this morning, or this afternoon, it was really surprising at how few genes there are. Yes?
So we're skipping the first 7 slides.
We are?
That's slide 9.
Then we'll say yes. We'll pick it up from here. Okay. Wonder how I did. Okay. The first 7 slides are an introduction. All right. And I think it's just kind of the general theory of behind what's there. Okay. You'll get the gist of this as we go. So this whole idea was, it was really surprising that there's so few genes in the human genome. But it wasn't until then the discovery that so many of these genes have these alternative splicing patterns and such, that maybe cells, in fact, can make 100, 150,000 different proteins in human cells, but they only need 25,000 genes to do it. Okay. The other is homologies, as whole genomes were sequenced. So kind of as a prelude to this, probably back in the 80s, probably, Carl Wu was kind of shaking up the field of systematics and phylogeny. In other words, the area of biology where you sit around and you argue whether this individual belongs in species A or species B, species C, and this was all built around morphology and such. So Carl Wu said, nah, let's do some molecular biology since we can do this now, and he started sequencing some of the ribosomal RNA genes and started developing phylogenetic trees based around those similarities, and there were a lot of inconsistencies between the phylogeny built around the morphology of the organism and these molecular tools. But on the other hand, he was only looking at a very select group of tools. I mean, if you lived here in South Florida and you haven't lived anywhere else, you might think this is the way the rest of the country operates. No, this is a different culture down here. Okay? I lived in Northern California. I love California. I love it down here. But they're two very, very different cultures. But now we're looking at whole genomes. So now you're looking at the whole story, making these comparisons. And what we find is between individuals, if unrelated individuals, the genomes are about 99.5% identical. So we only have about 0.5% variation between one individual to the next. That still translates to 4 or 5 million nucleotides, but it's a very, very small amount. Our greatest homology is to the chimpanzee among the primates. The homology there is in the range of about 96 to 98%. Later on, I'll talk a little bit about, instead of thinking so much about what's the same, what if we look in the areas of what's different and see what interesting things we can find there. If we get down into the mouse, one of our major models for biomedical studies and such, we got about a 90% homology. And there you find that about 75% of the mouse genes actually have a functional equivalent in humans. We get down into, with cows, about an 80% homology. With fruit flies, we're around 60% fruit flies and chickens. And still about half of the fruit fly genes have functional homologies to humans. A whole new, yes?
Do you know if the dark matter genes are included?
Not in this. Right. So that's a good point. The good point here is this doesn't include the dark matter, this junk DNA, because that's just, that information has only been out. Those papers were published in September of 2012, so they've only been out for about 3 years. So this would be among that's actually a very good point. It'd be among the genes. I would say this would be limited to really the genes that are transcribed into proteins and not the microRNAs and such. Okay. What's coming out of all of this, because of the changes in the technology and the speed and the cost of sequencing and such, is a whole area called metagenomics. So this is a very large-scale analysis of large numbers of genomes. It really started in the area of microbiology, really in microbes and thinking about communities. So we've known for a long time that we really only know how to culture a very small fraction of all the microorganisms out in the world. And up until now, if you couldn't culture it, if you couldn't purify it, if you couldn't grow it up in the laboratory, you couldn't analyze it. What this is really doing is saying, let's look at a whole community. So let's say you take a swab from somebody's hand, and I don't know, maybe there's 200, 300, 400 different species. You know, I know you guys use all that hand cleaner and all that, but you're wasting your time. but that's another story. So let's say there's 300 or 400 different species of bacteria. The idea is, with the technology available now, you don't have to grow those bacteria in the laboratory. We can get enough DNA just from the sampling of just taking a cotton swab, and a moist cotton swab, and just running it over your skin. We can get enough DNA to do sequencing, but you're looking at the sequences of all the organisms that are in that collection. So this would be what is called really a very gene-centric approach. So you might say, for instance, okay, let's look at DNA gyrase. How many different forms of DNA gyrase do we have? And if we have 150 different alleles of DNA gyrase, we probably have about 150 different species there. But it's this idea of being able to now look at community interactions that we didn't have the tools in place to be able to do this. And a lot of this metagenomics, and I'll give you a couple of examples in a few minutes, is now extending out into the human genomes, because now we can sequence. We'll talk about a project that looks at we can now sequence, there's literally thousands of human genomes have been sequenced now, so we can start looking at that kind of data. So this is a whole new area of research, a whole new area of potential careers for those of you who might decide you don't want to live with your parents for the rest of your adulthood. This is going to moving into personalized medicine. I think there's a lot of good career opportunities that are going to come from this type of data mining, data analysis of these DNA sequences. Which was, for instance, what started out was called the 1,000 Genome Project. The goal was to sequence the genomes of 1,000 individuals. They got a little carried away. The project ended up with the sequences from 2,553 individuals spread out over 26 different populations around the world. So the rationale is, if I want to know what genes are involved in cardiovascular disease, I need a large number of genome samples from people without the disease and from people with the disease. So by being able to compare large numbers, the genomes from large numbers of individuals, we can start to get a feel for what genes are important in a certain disease process or any other question you really want to ask. What also came out of that is nobody's perfect. And on average, it looks like the average person, about 1% of their genes, about 250 to 300 or so, actually are mutated to the point of being non-functional. So nobody gets to really be the wild type anymore. Okay. The other area that this is being used in is actually bringing back an old area of study. So this was an area called pharmacogenetics. So the field itself goes back probably several decades, and it's just this question about, why do different people have different responses to the same drug? But it kind of fell back in importance because you could ask that question, but there were no tools to really get you at that question. Well, now you can do that with this type of metagenomic analysis. So you can begin to look at the genomes of individuals and compare their response to a drug versus what their genetic pattern looks like. So it's my understanding that the FDA is using this now in clinical trials. So I mean, when you put your drug through clinical trials, you're not ever going to get 100% positive responses. I'm not sure what the number is. My guess is, I'll bet if you get 30%, it's probably quite good. So you're going to have those individuals who have no side effects but no response. Hopefully the ones that have a response, or your stock's going to go through the floor, but they don't have any side effects. Or those that have a response, but they also have side effects. But if you can do this type of detailed genomic analysis on the participants, you can build up a database that says, so in the future, let's say you're a physician and you're treating a particular disease, and you tell your patient, well, there's 3 approved treatments for this disease. Instead of making it personal and just saying, well, in my experience, A works the best, we can look at your genetic profile and say, based on your genetic profile, you'll probably have the best response with treatment B. And if that's for cancer, time is everything. As a colleague of mine said, your survival of cancer depends on who you see first. So this should help. But as I said, this gets into this area of personalized medicine, being able to look at individualized genomes and making up and deciding on a treatment regime based on that particular individual. Not up here, but in the same category, an area I really hope turns productive is immunotherapy, or this approach of saying, okay, why doesn't this patient's body recognize their cancer as being foreign? And now maybe we can reprogram their immune system to recognize it as foreign. If you can do that, if that's successful, I think cancer will be just another treatable disease. But it involves a lot of genomics, a lot of genetic analysis, and really, again, falls in this area of personalized medicine. Yes?
There was a three-month-year-old who was diagnosed with leukemia, I think. And I don't know if it was her blood or someone else's blood, but they genetically engineered it to specifically like attack and have like tumor cells like turn on each other. And like she's 3 now and has like no
Great.
symptoms of cancer. And like it didn't use a pill or anything, so it didn't harm her liver or anything like that. It was
Yeah, I think this is an incredible approach. The pioneer in this field was awarded the Nobel Prize about 2 or 3 years ago. But he also had pancreatic cancer, and he was trying this on himself. And the story goes, this is, if I remember the story correctly, and I should have remembered his name, but I don't. He passed away on a Friday. The Nobel Committee decided he would receive the prize over that weekend. They made that decision. Tried to contact him on Monday, found out he had passed away. And as I think I talked about with Rosalind Franklin and Watson and Crick's Nobel Prize, the rules specifically say you can't receive the prize posthumously. But there is a rule that says if you pass away, since a lot of these people are old when they get the prize, okay? If you pass away between the time it's announced and the actual ceremony, then you still keep the prize. And so the committee decided, since they didn't know he had passed away, that he would keep the prize, his family would get the money. It was $500,000 or something at the time. And he's in the books for having it. But no, I'm glad to hear that because it's not an area I work in. It's an area I know Dr. Hartman's very interested in. But I think as much as I love the biotechnology and the genes to drugs and all of this, I don't think any of the drugs are going to get every last cell. But I think this could.
They said the success of that one procedure would like just blew the roof off of like people actually want to start doing that.
Yeah.
It's really got people.
This could be a tipping point. And I think if it does, then I think cancer is just going to become another treatable disease. So yeah, I mean, there's so much of what can be done with this now that this information, now we can get to this. Here's just another example. You can do a genetic testing to look to see what's the probability that you'll be able to metabolize certain of the common medications. This is why the side effects are there, is because you can't metabolize these drugs in certain ways. This could be a way to sort of sidestep some of the serious side effects ahead of time, or may even affect which drugs are used. The other area is synthetic biology. Inventors is leading the way in this area, and that is creating whole new. The ultimate goal would be creating novel organisms from scratch. So he's basically trying to build a one-celled organism where he's developing the genome synthetically. Other possibilities are in genetic modifications, making microbes do new types of tasks, such as detoxification of hazardous waste, which is interesting, something I don't hear about too much, but there was a very active field called bioremediation, which was basically using microbes to degrade toxic compounds such as oil, hydrocarbon waste, and such. There you're looking for natural isolates. Like if you wanted to find bacteria, well, a former microbiologist in the department thought he had isolated some bacteria that could degrade polystyrene. Where did he probably find those bacteria? At the dump, exactly. These microbes are incredible little biochemical factories, so he isolated it from some samples he found at a landfill, and then his dumbass graduate student lost them and didn't culture them properly. Don't worry, he didn't graduate either. But But here now, this is sort of, one is naturally occurring isolates. This is thinking, well, no, this is a little more genetic engineering out there. Of course, we got to be very careful here. We don't really know where we're going in some of these areas. So this type of modifications and such are highly regulated. But it's coming. This is the future. There's kind of a collective term that really wasn't even part of our lexicon until the mid-1990s called functional genomics. And that is, it really is, how do we take this, all this DNA data that we have and actually data mine it to get something useful out of it? And it's also given rise to lots of ohm words. So you have the genome, which is all the DNA sequences in the organism. Now you have the transcriptome, which is a full collection of all the transcripts or RNA molecules in the cell. Then there's the proteome, which is a collection of all the proteins in the cell. Okay. But the main challenge here with functional genomics is, even though we now know there's about 25,000 genes in the human genome, we actually only know the function of a small percentage of those. Even E. coli is extensively, as E. coli has been studied, there's about 6,000 genes in E. coli. Half of those genes we still don't know the function of. So what happens is you're doing experiments in these sort of reverse genetics type of experiments, and you find a DNA sequence that's interesting. It looks like it codes for a protein. What does that protein do? I don't know. Nobody knows what that protein does. So functional genomics is really a set of tools that tries to look at other DNA sequences and help you predict what the function of that protein might be. Now, a very important point here is this type of data mining, this computer-based research, while it's critically important, does not replace wet labs. You have to go and validate. It helps you refine your experiment, make it less of a fishing expedition, but you still have to go into the lab, and many times you find out that, in fact, experimentally, it doesn't support your initial conclusion, but that's how the process works. Okay. So a lot of this is based on this trying to predict protein function. So if, because of evolution, again, I think the state still allows me to talk about it, although I'll only stand on my soapbox for one moment. If Carson gets to be president, I don't know if I get to talk about evolution anymore. Boy, let's start that firestorm. No, let's not. Okay. Back to what we should be doing. So part of it is you got this new DNA sequence, and you can predict the protein sequence. At least you can get the primary amino acid sequence. Now, software is getting really good. The algorithms are getting much better at predicting how that protein might fold up and what it might look like in 3 dimensions. Then you can start looking for motifs on it. And then you start seeing that, oh, look, this looks like a zinc finger. This looks like a leucine zipper. This is probably a DNA binding protein. So what you do is try to look for structural similarities that are predicted for your protein compared to known proteins, and then that'll help guide you in the laboratory as to what experiments to do to really prove what the functionality of that protein is. Okay. So just to help make things complicated, we can talk about orthologs and paralogs. All right. This is actually really important. I mean, this is how it's important because this is how I justify to the NIH why studying my particular problem in a fruit fly will actually contribute to understanding a human health condition. And that is, we have functional similarities between proteins in these different species. Okay. So orthologs are homologous genes from different species that arose from a common ancestor. So you're going to find them in different species. Paralogs are homologous genes within the same organism. So the globin genes would be which ones? They'd be paralogs where they are duplications, and then they begin to diverge out and take on different functionalities, but they're within the same species. Okay? And the whole idea is by doing this, sometimes even by sequence homology, you can begin to predict that this human protein is probably very similar to this Drosophila protein. All right? or jumping ahead to the last lecture, just to show it as an example, when people are isolating genes involved in development in Drosophila, they noticed that at the beginning of every one of these proteins was a very conserved 60 amino acid sequence that they called the homeodomain. Well, at the time, mouse genetics was not nearly as well developed, so there was really no way to genetically find these developmentally important genes in a mouse. And the idea was, well, what if mice also have this homeodomain associated with all of its developmentally important genes? So let's start searching the mouse genome for proteins that have this 60 amino acid sequence or something very similar to it. And that's exactly what happened, and it helped. So that was a way, by using data from Drosophila, you could bridge over to finding the analogous genes in the mouse, and from there over into the Cox genes and the Hox genes in humans. Okay, so here's just an example from, this is probably from zebrafish. The idea is just that we have the paralogs, which is just gene duplications, and then they take on different functionalities. And then after a speciation event, those analogous genes in these two different species would be orthologs. Okay. So let's see if we can sort all that out. And here we go. No, this looks like it only has to take a minute. Okay, I'll go to 1 minute. All right. So what's the answer? Okay. Yes. Oh. Okay. All right. So the next few slides really try to give you some little bit of an insight of how you can take information from the human genome and really begin to think about this in terms of treating diseases. So, in the past, many treatments for diseases were either trial and error, as we talked about before, let's say using nucleotide analogs just to kill cells that are rapidly replicating, but that isn't really targeted to the cancer cells or to the virus, because it also hits certain cell populations in the patient, and then you have these side effects, and it may be efficient, not efficient. So let's try to do this in a more surgical way. Let's try to find a particular gene to target for this particular disease that we're working on. So now these drug approaches and treatment approaches are really very tissue-specific, cell type specific, and actually targeting in on a specific gene. So this is this whole sort of big, coming under this big umbrella of bioinformatics. And so, for instance, most of us use the GenBank DNA database from NCBI. This is the National Center for Biotechnology Information. It is freely, it's publicly accessible, and so now you use something called a blast search. Means you put in your DNA sequence and you're asking the computer to compare it against the literally billions and billions of nucleotides in the database. The students get all anxious because it takes 2 to 3 minutes to get the response back. Well, this really wouldn't be available without the internet, because this is running on some kind of crazy supercomputer, and most universities even don't have the computer capabilities to do this. And the reason it takes 2 to 3 minutes is because you're just sitting in line along with everybody else around the world who's waiting for their responses to come back. So this is the so-called data mining. And the general idea is, let's look at the expression of genes in this normal cell and the expression of genes in this tumor cell, and see if we can find a gene whose activity is either upregulated or downregulated in this tumor cell. It's different, and maybe that can be our target. Now, let's say this gene is upregulated in the tumor cell. Well, if I could interfere with either the expression of that gene or the protein product, I might actually be able to affect the growth of that tumor. It may begin to regress, or maybe it might even just be a biomarker for a very early marker, because mainly our success with cancer comes from the earlier it's detected, your better chances of curing it. So to be able to do that, and do this in the laboratory, we had to be able to see how this gene of interest is acting. So we typically use something called a reporter gene. All this means is we're just going to use a gene whose product is easy to follow. It's either going to be fluorescent or it's going to create a color. So for instance, two of the most common ones are the green fluorescent protein, which actually functions even in living cells, or some assay with our old friend beta-galactosidase or the LAC-Z gene. There's lots of substrates that, for instance, XGAL is a substrate. When it gets cleaved by the beta-galactosidase, it forms a dark blue color. We want some sort of reporter system. We want to be able to know, is our gene turned on or is our gene turned off? So here's an example of it. Here's a case. So in this case, we're using a transgene. This one happens to be in a tadpole. And it's got the green fluorescent protein. Wherever you've seen this green fluorescence, it's showing where beta tubulin is present in the brain. So this is just one example, but you can get very high resolution, almost cell-by-cell resolution of where these proteins are being expressed. There's some very elegant, yes? What? Oh.
So could this be used, say, if you were to synthesize bacteria and want to use it to identify a disease?
If I'm following what the question is, could it be used, let's say, in genetically modified bacteria?
To identify disease in a sample from somebody. Like maybe if it encounters something and it could...
Along those lines, I think it's been used in environmental studies. So basically looking for toxins. So if this particular toxin might be present, it may these may have set the system up to be activated. The gene would get activated in the presence of toxin X. And if you start seeing that, so yeah, I mean, the potential is there. I don't know in the example you're giving me whether anybody's done that, but the potential is there. It's really just kind of limited by your imagination. How do you like that for an incomplete answer? That's called, I really don't know, but it's a really interesting, but it's got, I mean, these areas are moving so fast, I can't, I can't keep up in these areas. So that's what your job is. But it's just the technology is proliferating to a point where you have these tools to be able to tackle so many questions that you just simply could not answer before. Okay. Now, usually what happens, so let's say you find this. Let's say you found your target gene and it makes protein X, and you think that this protein X this gene is being expressed at 5 times its normal level in these tumor cells. So it's 5 times as much of this protein. If I could knock that protein down, if I could inactivate that protein and get it back to more normal levels, maybe I might get regression of the tumor. So the easiest way to do that is to look for a small molecule that can bind irreversibly, let's say, to one of the substrate binding sites or the active domain of this enzyme and inactivate it. Now, again, algorithms are improving to begin to predict what small molecule might work, but it's still pretty much a trial and error method. So this goes into what's called high throughput screening. So a lot of molecular biology today is, because of the technology available, is high throughput just means I'm running huge numbers of assays on this. So here's an example. Scripps, Florida has a robotic system called Calypso. And they have a library of over 1 million small molecules. So the idea is the molecular biologists have set up the assay. They have their reporter gene, so they know when the enzyme's active, when it's not active. And so all we got to do now is run an assay with each of these 1 million compounds, and we'll see if we can find one that when it binds to that protein, it inactivates the enzyme. Okay? So you take that to a graduate student and you go, how would you like to do 1 million assays? Okay. No, you don't get too many takers on that, and besides, it'll take several years to do it. So this is a high-throughput robotic system. So there's a lot of engineering, computer science in molecular biology today. And once the whole assay is set up, it might take a year to develop the assay. But once it's set up, the robots can run through these assays, and I've been told in about 2 days. Plus, they don't get health insurance or any other benefits. So that whenever you see high throughput, it just means it's something it's an experimental procedure that's going to probably involve tens of thousands or more assays. Another approach is something called microarray analysis. Up until about the mid-1990s, there really wasn't a good way to study more than 1 or 2 genes at a time. It just wasn't technically possible, and people made entire careers out of studying 1 or 2 particular genes from Drosophila and such. Or if you want, and you could use that kind of approach for human health diseases for something like cystic fibrosis or Huntington's disease, well, there's just one target gene that's involved. But if you think about things like cardiovascular disease, diabetes and such, it probably involves a large number of genes. And how do we actually look at large numbers of genes? And so what came out of this was microarray analysis. So just very briefly, it came from computer chip technology. So it's a glass chip where microscopic dots of DNA are imprinted onto the chip. So that's what you really have here. Each of these little dots is a different DNA sequence, and then it was probed with RNA from particular tissues and such. Each of the different colors comes from different experimental conditions. The real take-home message is, using this approach, you can screen the gene expression of the entire human genome in a single experiment. So you can look at changes in gene expression of the 25,000 genes in a single experiment. This used to be very expensive. A company called Affymetrix has the patent on it. We had one of their gene chip machines here. It was at $500,000 when we first looked at it. We finally got them down to $250,000 for it. Turns out it's a very expensive toy. It costs about $100,000 a year to maintain it. We don't have it any longer. But doing one experiment with the human genome initially could be anywhere from $10,000 to $20,000. Now it's probably down in the realm of $1,000 or less. It's a lot of money if it comes out of your pocket, but it's doable off of a grant. So let me just say what we want. I just want to give you one example of how this worked, and then we'll call it a day. So to show you how this has been used in this type of analysis, so this was a study of breast cancer in young women. And so these were young women with primary tumors, and they looked for the gene expression pattern of all 25,000 genes in the human genome. Then they followed the women for 5 years, and in this case, 34 of the 78 women, the tumor metastasized and spread through other parts of the body. 44 of the women were disease-free. When they looked at the data, what they found was, and everybody knows about BRCA1 and 2, but they found 70 genes whose expression pattern, out of that 25,000, this collection of 70 genes, their expression pattern could accurately predict whether the disease was likely to metastasize or not. So it's just preliminary results, but it's a little example of how this type of microarray analysis and this meta-analysis of large numbers of genomes could impact human health going forward. All right. So let's use that as a good stopping point. So we'll pick this up on Wednesday.